kind introduction. It's a pleasure to talk with you guys today. I wish it could be in person, but perhaps one of these days. So let me go ahead and share my screen. Can you guys see that okay? Great. Okay, so as Jiv mentioned, today I'll be talking about some work that I and my collaborators and now my new lab here at Dartmouth have been doing over the last few years, trying to advance a framework for using naturalistic stimuli to look at individual differences. And just by way of background, so I joined Dartmouth in July 2020 in the height of the pandemic. I'm finally in my office, as you can see, but I'm still working on filling out the shelves. So it's really been an interesting year or so. And so I was hoping to have a little bit more new work to present today, but that may have to wait until next time. But I'll try to give you sort of a broad picture of the lines that we're thinking along and some of the ideas that we're excited about. So I'd like to start off this talk by presenting this quote from Plato, who wrote more than 2 million years ago now. Sorry, 2,000 years ago. It is early in the morning. No two persons are born exactly alike, but each differs from the other in natural endowments, one being suited for one occupation and the other for another. So, you know, for anybody that lives in society and interacts with other humans, it's sort of no secret that we're all different in certain ways. I'm really interested in the neural basis of these differences. And I think that there are 2 main reasons to care about individual differences when we talk about human psychology and human neuroscience. And the first main reason to study individual differences is that I believe it could lead us toward a deeper understanding of cognition more generally. So after almost 3 decades now, really, of functional MRI, we have these group maps that are pretty well worked out. You know, so these are data-driven meta-analyses from neuroscience.org that's pulling from hundreds or thousands of studies. But, you know, for most human imaging researchers, if I sort of say, you know, things like working memory or language or social cognition, there are sort of these canonical maps that we might think of the brain areas that tend to be most involved in these different processes. But of course, we also know that these group averages are made up of lots of individuals whose maps may look slightly or even substantially different from one another. And I think these differences can be important, and these differences can manifest in lots of different ways. So what this is telling us really is that there's a degeneracy between the cognitive process and the individual brain implementation. So different brains can implement the same process in different ways. And these differences could, you know, relate to things like performance on the task. And they could relate in linear or monotonic ways or even non-monotonic ways. But even holding performance constant, these differences could reflect things like differences in cognitive strategies or styles. There could also be within subject changes, like with learning. And then all of these potential reasons for these differences could map on to different features that we could pull out of the brain data itself. So we could see univariate or multivariate relationships with these patterns. We could see relationships with activity, connectivity between regions, or really any other feature that we could pull out from the brain data. And so the point here is that I think by getting a handle on why and where and how we observe these differences, it gets us closer to a more rigorous understanding of cognition. So if we can map from a cognitive process in an individual brain to a behavioral output, that is a more nuanced and more rigorous understanding of how individual brains actually implement these cognitive processes. The second main reason I think we should care about individual differences is that it could potentially give us insight into mental illness. And so the traditional approach to studying mental illness was always to essentially take traits that are by and large distributed normally in the general population. So this might be something like trait anxiety, for example. And then typically when we try to study brain correlates of trait anxiety, we take this kind of categorical approach where you know, we call everyone on the extreme end of the distribution of patient and everybody that's sort of below that threshold of control. And then we might extract some feature of their brain and compare group means. And you can see in this toy example that even though, you know, the patients have a slightly larger amount of this variable on average, there's also a lot of heterogeneity within each of these populations that's not being captured by this approach. And so the approach that we take in our lab and that many other people are also taking these days is to preserve the dimensional nature of this trait. So to treat something like anxiety, for example, as the continuous variable that it really is. And then place everybody on the same axis. So now we can map this continuous relationship between something that we're extracting about the brain and levels of this trait. And I think even in certain psychology experiments where we don't necessarily have access to patients within the experiment itself, doing things in this way can still potentially give us insight down the road into illness. So even if, for example, we only have this part of the distribution represented in our sample, by parameterizing this curve here, it could still potentially give us insight into what's happening at these extremes. And the hope here, of course, is that one day this type of work or this type of approach could more quickly lead to translational tools based on neuroimaging for clinical or other real world situations. So with those two motivations in mind, why do I think naturalistic tasks are a good place to look at these differences? I think things like movies and stories that sort of more closely mimic real life, let's say, than the traditional tasks that we often use in psychology, they just leave much more room open for different interpretations, different reactions. People are bringing different experiences to the table, different memories. You can kind of see in this toy cartoon here. It provides a really interesting test bed, so you can give everyone exactly the same sensory stimulation, but they're going to have different interpretations and different reactions and feel differently. And so I think from both a theoretical and also sort of a methodological standpoint, which I'll get into later, looking at these individual differences during naturalistic tasks. And by that, for purposes of this talk, I just mean tasks like watching a movie or listening to a story in the scanner. I think these are really interesting and powerful ways to pull out these differences. So with that background, I'll give you a brief outline of what I'll talk about today. In the first part of the talk, I'll be asking and answering this question of, can we detect stable and meaningful individual differences during naturalistic stimulation? And I'll get into what I mean by both stable and meaningful. On the second part, I'll talk about a study we ran to sort of see if we could design a bespoke naturalistic stimulus to draw out variation of interest. So targeting a specific trait and actually tailoring the stimulus content to be able to draw out individual variation in that trait. And then finally, I'll spend a little bit of time at the end talking about how we should model individual variability during naturalistic stimulation. So beginning with this first part, can we detect stable and meaningful individual differences during naturalistic stimulation? For this first part of the talk, I'll largely be relying on functional connectivity as an analysis technique. And I know many people are probably familiar with that, but there's a lot of ways of doing functional connectivity. So just to get us on the same page, in the functional connectivity approach that I and my collaborators often take, we take the entire brain, we carve it up into a set of predefined nodes. The atlas that we use has 268, if anyone's curious, but essentially these are areas covering the whole brain, including both the subcortex and the cerebellum. And then essentially for any pair of these nodes, we just look at the Pearson correlation in their signal time courses over the course of a scan. And in this case, maybe we observe that there's a strong correlation here. So we're not necessarily inferring causation from this or any other directed relationship, but we are able to say that these regions are similar in their activity fluctuations. And if we do that for all possible pairs of nodes across the brain, we can construct this symmetric functional connectivity matrix. And we can do this for an individual subject, and we can do it at rest, but importantly, we can also do it during cognitive tasks or naturalistic tasks. So for the first part of the talk, this is sort of the basic unit that I'll be working with. And just to briefly kind of overview the study that first got me interested in individual differences and functional connectivity as a way to study individual differences. This was a result that we got a few years back where we were using data from the Human Connectome project. And essentially we wanted to know, how stable and unique is that matrix across different people. And so in this data set, we had 126 healthy adults. They were 22 to 35 years old, actually a pretty homogeneous population. And interestingly, there were 50 sets of twins. So 100 of the 126 people in this data set actually had a twin in the data set, which served to make our problem actually even harder. Because our problem was this, basically we had, if you're familiar with the Human Connectome project data, there's several different functional scans over the course of 2 days. There's rest and then a few cognitive tasks, kind of traditional tasks. And really, we just were asking the very simple question of, if we calculate one of those functional connectivity matrices for every subject in each one of these conditions, can we then match a matrix from the same person across different scans, separated by at least a day or so, and also different conditions. So rest, not only to rest, but also to things like language and emotion, et cetera. And long story short, we observed that we were able to do this with a pretty striking degree of accuracy, suggesting that there is, to some extent, a stable individual signature present in these matrices that's there regardless of what people are doing in the scanner. And we looked to see, where in the brain the most identifying information was. We found that it was most closely related to these two networks that I'm highlighting in teal and purple. So, called in various settings frontoparietal networks. You can see it's mostly concentrated in higher order association cortices. So this is sort of where we might expect that information to be coming from. And indeed, that's where the most individually distinct connectivity tends to be located. So this was kind of an interesting result, but as I told journalists at the time, we're very excited about this paper. We don't need to necessarily put someone into an MRI scanner to know who they are. We can tell who they are by looking at them, for example, or by looking at their actual fingerprints or their DNA or things like that. So really, the primary interesting thing from this type of work, for me at least, was not just can we tell who someone is, but is there meaningful information embedded in these functional connectivity patterns that we can use to predict some sort of behavioral trait or aspect of people. And so as part of this first study, we were able to show that using a pretty simple machine learning approach, we were able to predict people's score on a fluid intelligence test using just their resting state connectivity. And so that was exciting to us because, again, it was suggesting that not only are these connectomes stable and unique, but they also contain some kind of meaningful information about how a person can operate in the real world. And similar to the fingerprinting results, most of the predictive power was coming from these two same networks. So with all of that in mind, we thought that naturalistic, so I guess around this point, I started to get really interested in just naturalistic scans in general. So things like movies and stories and how we can study the brain under those conditions. And we actually thought it would be an interesting challenge to these fingerprinting results because we knew from a separate tradition of work, first pioneered by Uri Hassan and then extended by many other people in the field, that naturalistic tasks tend to really synchronize people's brains. So in other words, if you and I are watching the same movie, our brains are actually looking pretty similar and probably much more similar than they would if we were just resting or even if we were doing sort of a more traditional cognitive task. So movies are really rich and engaging. They drive lots of different brain systems at the same time. And so we thought this would be a really interesting challenge for our fingerprinting paradigm to see if we could still identify people or separate people from one another under these conditions where people look pretty similar. And so lucky for us, we had a collaborator at Yale, Tammy Vanderwall, who had collected a data set that we could test this in. This was a smaller data set than HCP, but still large enough to start to look at this question. So these were healthy, young-ish subjects, and basically they came for a test-retest design where across 2 sessions, they did 3 different types of scans. These were all counterbalanced in order, but basically there was a rest scan, and then there were 2 different types of movies. Maybe some of you are familiar with this Inscapes paradigm. I'm showing examples from the movies on the right. So Inscapes is sort of this dreamy, abstract, sort of computer-generated animation that's really visually pleasing, but doesn't have much of a story, or it doesn't really have any concrete objects that you can recognize. And that was in contrast to, sorry, I'll play this over again, this clip from Ocean's 11, which is basically on the other end of the spectrum. So lots of dialogue, fast-paced, sort of typical Hollywood-style movie. And so we had these 2 very different movies plus resting state. These both had audio too, by the way, which I'm not playing for you now. And our goal was just to see, how well does fingerprinting hold up under these conditions where we expect people to look much more similar to one another. And what we found was that movie watching preserves, and in some cases numerically even enhances identifiability. So what you're seeing here in this matrix is the accuracy of identifying someone across 2 different scans. So for example, when we try to match rest 1 to rest 2, we see about 91% accuracy. When we try to match rest 2 to rest 1, we see about 80% accuracy. So overall, these numbers are all well above chance. If you look down the diagonal, you can see these are how well we do when we try to match people across matched pairs of scans. So rest 1 to rest 2, Inscapes 1 to Inscapes 2, et cetera. And it's difficult to statistically quantify this because we really only get one number per study, because this is an overall accuracy of how many people we get right essentially in the data set. But you can see that if anything numerically, we're actually getting up to 100% accuracy with one of the Oceans pairings. And that's actually the movie that does, if you look at intersubject correlation, for example, does make people look the most similar. So somehow we're making people look similar, but we're still preserving something about the functional connectivity matrix that is stable and unique to each person. And then these off-diagonal elements too, where we're matching across different scan conditions, are also quite high and well above chance. So this was sort of an encouraging proof of concept for me that we can use naturalistic stimuli to study individual differences. You know, we're not completely saturating or wiping out this individual signal, even though we see these strong intersubject correlations as people watch the same movie. There's still something about the individual that's there and that's stable and that's unique to them. But of course, I told you in the fingerprinting study that you know, I think the primary motivation for doing these types of analyses is not just identification for its own sake, but rather seeing if we can use brain data to predict behavior. And as we were exploring this study or these results in Tammy's data set, there was also a lot of evidence coming out that actually task-based connectivity can do a lot better for predicting behavior than rest connectivity. So those results that I showed you for fluid intelligence were all done based on rest, but it turns out that even in the same subjects for predicting the same exact behavior, if you feed your model connectivity data from a task, really any task, you do better for predicting behavior than if you feed it resting state data. And this is a paper from my former lab where they showed this very nicely in 2 different data sets. So both the Human Connectome Project and the Penn Neurodevelopmental Cohort, which is a children and adolescent study. And basically on the x-axis, you see the scan condition that the matrices are calculated from, and on the y-axis is how well you do for predicting fluid intelligence. And in both of these data sets, these are rest out here on the all the way on the right and here too. And so even though rest gives you significant predictions, it turns out that tasks on the just across the board do better for this. And there's a few reasons I think that this might be the case that I'd be happy to talk about later if people are curious. But essentially, this then led us to ask the question, well, okay, if we can still identify people while they're watching a movie, and at the same time, we're seeing this evidence that more traditional cognitive tasks are giving us better behavior prediction, what happens when we try to use movie connectivity for predicting behavior? And again, luckily for us, the Human Connectome Project had a separate data set. This was the data that was collected at 7T that was going to allow us to ask this question. And so we were able to directly pit movie watching against resting state for seeing how well we could predict behavior. So the 7T data collection was a smaller one, so we had access to, but still, you know, fairly large enough. We had 176 healthy young adults who took part in this study, and we took 2. So if you're familiar with this data set, you know, there's tons of phenotypic and behavioral variables. We wanted to focus on 2 broad domains. The first one was cognition, and the second one was sort of socioaffective traits. And so we did some principal components analysis to kind of boil down some of families of measures into a single score for cognition and a single score for socioaffective. And I'm happy to talk more about that. But, you know, we didn't have strong theoretical motivations going in. We kind of wanted to explore movie-based prediction for these 2 broad domains. So I'll be talking about predicting both of these as target scores in the next few slides. And so the structure of this data collection effort was also 2 days of scanning. There were actually other days sort of in between, but we're focusing on sessions that have both rest and movie watching. And so the movie scans were done in 2 15-minute scans. And the important thing to know here is that it wasn't one continuous 15-minute movie. It was actually several different shorter clips. So clips on the order of like 2 to 4 minutes separated by 20 seconds of rest. And though that's what comprised these individual movie runs. And so just to get right into it, basically we took our connectome-based predictive modeling approach and we applied it to both the resting state and the movie watching data to try to predict these two scores, so cognitive ability and socioaffective traits. And what we see, so what you're looking at here in this graph these are predictions accuracy of our model. So on the x-axis is the run that we're feeding the model, and in dark gray is our resting scans, in purple our movie scans. We ran the model about 100 times just to get a distribution of accuracies. So that's what these colored dots are here, and that's compared to a null distribution that we generate via permutation. And so you can see, and then on the x-axis is essentially correlation between observed and predicted scores for, in this case, cognitive ability. And so what you can see here is that in the first session, we're getting significant predictions from rest, which is good. That's sort of replicating our previous results. But it turns out that the movies, and especially this movie 2, which I'll talk about a little more later, do a lot better for predicting this cognitive ability. So again, exact same subjects, exact same target score, but just by feeding it data for movie watching, we're doing better than if we have people simply resting. The results from session 2 were a little bit weaker. This was after several days of scanning, so the data quality, I think, suffered a bit, but you can see kind of the same overall pattern. So predictions from rest for cognitive ability were actually not significant here, but predictions from the 2 movie runs were both significantly above chance as well as significantly different from rest. And so that was encouraging on the cognitive ability side. On the side of the socioaffective tendencies, these turned out to be much harder to predict overall, and this is similar to what others have found. One major difference between these 2 domains is that the socioaffective tendencies were all self-report based, whereas the cognitive ability was all based on performance on behavioral tests. So these measures are just sort of trickier in general, but to the extent that we could predict this score at all, we were only able to predict it from this movie 1 run in session 1. And I think the session 2 predictions for this score were not significant, which is why I'm not showing them here. But overall, this was some encouraging evidence that we can improve over rest by using movies for behavior prediction. And as I mentioned, the interesting thing about these movies is that they were actually comprised of these several different individual clips. And so our next move was to ask, basically, does the content of the movie matter? Or, are there some clips that do even better than others for generating these predictions? And so we went back and calculated connectivity matrices from each of these clips individually. And that's what you see along the x-axis here. And then same thing on the y is prediction accuracy, and that's always compared to a null distribution. And you can see some really interesting, I think, variation between how these clips are doing. And again, each of these clips is only about 2 minutes. We actually truncated these so that they were all exactly the same length to remove any confounds there. So I think these were only about 2 minutes, maybe a little over 2 minutes. And for many of them, we're able to generate significantly accurate predictions. Not all of them, but interestingly, some of them are doing really, really well. So even much better than other clips. You can see these 2 that kind of jump out from movie 2. These are clips from The Social Network, if you're familiar with that movie about Mark Zuckerberg and Facebook. And coincidentally, also Ocean's 11, which was the same clip that, not the same clip, but the same movie that Tammy had used in her study. So if you're familiar with these movies, you know that these are both sort of fast-paced, kind of Hollywood style, lots of characters, lots of social dynamics, and I'll come back to that in a bit. But essentially, these results are telling us that yes, content does matter. Not all movies are created equal. There's some really interesting differences in how clips perform for predicting specifically this cognitive score. When we do the same thing for emotion, again, we see that overall the sort of socioaffective emotional tendencies are overall harder to predict. So there are fewer clips that are giving us significant predictions. But to the extent that clips are doing so, we see these same 2 clips doing well for predicting emotion. We also see this pockets clip here. This is sort of from this documentary where people are showing the camera what they have in their pockets and talking about why it's meaningful to them. So anyway, so point being that there's some interesting variation across clips in terms of how they do for predicting these 2 scores. And, in looking at which clips did best, what sort of started to occur to us was it does seem that these clips that have a lot of just a lot of humans, a lot of characters, a lot of dialogue, a lot of intrigue in terms of what's going on in the social situation, tended to be doing better than clips with less of that type of content. And so we quantified this by just essentially counting or taking a percentage of the number of TRs that contained at least one face. So this is just a proxy, a crude proxy for social content. How many, what percentage of the time is at least one face on the screen? And if you correlate that with the median prediction accuracy from those models that we ran, you do see this positive correlation such that clips with more social content do better for predicting this cognitive score, which is actually interesting because, we're not predicting anything directly social here. We are predicting this sort of more general cognition measure. And I can come back to what I think that might mean, but essentially, these are just screen grabs from the best performing clips and the two worst performing clips. So you can kind of get a sense for the differences in content here. I mean, all three of these clips have, again, just a lot of interaction among people. These two clips are sort of more nature montages or kind of indie documentary style things where there's not a lot of structured character interaction. And so we thought this was really interesting. But, one sort of question with this is, well, is it just the case that the social clips are more engaging? And so they're doing a better job of capturing people's attention. And that's why, we're sort of seeing this boost in signal because people are just more aroused. They're just paying better attention. Maybe our data quality is better. By the way, we did do several checks into things like head motion, and there was some eye tracking data on these people. So it didn't seem like people were just sort of asleep during rest and awake during movies. There were no sort of broad strokes differences in data quality between the two types of scans. But still, we kind of wanted to dig into this a little bit more and see, can we tease out, is this just sort of a general engagement effect, or is it actually something specific to this social content? And we weren't, able to do this exactly the way we wanted to, just because we were working with the clips that we were given. But one thing that occurred to us to try to get at this question was to turn to the eye tracking data. So again, they had eye tracking data that was usable in about 2/3 to 3/4 of the subjects in this data set. And we reasoned that, maybe if a clip is just more engaging across the board, people should be more synchronized in their eye movements to that clip because their attention is being directed in the same way. And so we could use synchrony of gaze essentially as a proxy for how engaging a clip was or how well it was driving people's attention in the same way. And to try to quantify that, we calculated this sort of eye tracking ISC, or intersubject correlation measure, where essentially we're taking gaze position across time for every pair of subjects, and we're correlating that into a big subject by subject matrix. So basically, this distribution of values tells us generally how much people were looking in the same place at the same time. And we hypothesized that if this general engagement effect were accounting for our results, that mean or median synchrony of gaze position should correlate with prediction accuracy. We didn't actually find a relationship there. So the overall or the median synchrony in this matrix did not significantly relate to the prediction accuracy for that particular clip. However, interestingly, the standard deviation of eye tracking ISC values across subject pairs did relate to, or did sort of predict in a way, better prediction accuracy. So in other words, basically the clips that are causing people, some pairs of people have very similar gaze trajectories, other pairs of people have very different gaze trajectories. Those are the clips that do the best for predicting this cognition score. And so what that's suggesting, I believe, is that it's not necessarily clips that are just across the board more engaging for everybody, but it's clips that are engaging different people in different ways that are bringing out or enhancing these relationships with overall cognitive score. Excuse me. And so there's a lot more to unpack here. There's a lot more that we could do using targeted clip selection and different ways to quantify these things. But we felt that this was an interesting result, and we're gonna hopefully be able to follow up on this. So to outline this first section, which by the way was by far the longest section. So So can we detect stable and meaningful individual differences during naturalistic stimulation? So I'm going to say yes, in the sense that identifiability, or quote unquote fingerprinting, is preserved during movie watching. So that tells us that there's stability there. And also we're seeing that movie watching outperforms rest, actually in most cases, for predicting behavior generally. And so that tells us that these individual differences are meaningful. If anyone has any questions at this point or at any point, I'm happy to take questions during also. But if not, I will go on. So with these results, another thing that we had been interested in for a while and we're excited about was this possibility of actually designing a bespoke naturalistic stimulus to try to draw out variation along an axis that we care about. So far, basically everything I've shown you has been on these large-scale publicly available data sets, which are wonderful for discovery science, and I've benefited a lot from them. But it's also, you can't really do sort of targeted hypothesis-driven manipulations of different things. And so one really interesting thing about naturalistic tasks is this sort of, the big decision is always, well, what do you use as your stimulus? And this, we felt, presented an opportunity to really design or craft a stimulus that we thought could be targeted to a particular trait, and see if we could pull out some interesting relationships with behavior doing that. And so I'll tell you about a study that we did a couple of years ago now, and the trait that we decided to go after was trait paranoia. So this is an interesting one because we know that paranoia in its extreme form is related to or is a symptom of clinical disorders like schizophrenia and other psychotic illnesses, but paranoia also exists on a spectrum, even in the healthy population. And so we thought it could be really interesting to try to design a stimulus that some people would hear, and sort of depending on their baseline level or level of trait paranoia, some people might find it really suspicious or kind of threatening, and other people might get a more neutral or even positive read on the situation. And so we actually crafted an original short story to serve as the narrative for this, or as the stimulus for this experiment. And the reason you're looking at a jungle is because the story basically was about an American physician who goes down to the rural Amazon jungle in Peru to try to work at a health clinic there. And this was actually all done pre-COVID, by the way, but now it's sort of funny to think back on this. But basically, there's a mysterious illness that she can't figure out what it is. And at times, it seems like she may have been lured there under false pretenses, like maybe there's some kind of conspiracy. At other times, her interactions with the villagers are quite positive. And so the story kind of leaves off at a climax. You don't actually know what the resolution is. And the idea is that different people will hear this story and come to different conclusions or different interpretations. And so on all of our subjects, we collected lots of different cognitive and self-report scales, including trait paranoia. And then everybody came and heard the exact same version of the story. But again, the idea was that they would have different interpretations, and we were able to operationalize that in a few different ways. And so pivoting a little bit from functional connectivity, I've sort of previewed this a little bit, but one really nice thing about naturalistic tasks is that they give you this ground truth time course that you can use in your analysis. So unlike rest, where everybody is sort of doing different things at different times, with a naturalistic stimulus, you do have the fact that everybody's getting the same exact sensory input at the same time. And so you can use techniques such as intersubject correlation, which just to give a basic intro to this, this is the idea that if two people are exposed to the same information at the same time, if I look at the same region, voxel or larger region in two people's brains, and I see that there's a correlation in the signal in that region, I can infer that that region is somehow responding to the stimulus. So the stimulus is synchronizing that brain region across people. And so what we wanted to do here was extend this a little bit to ask not just what regions are responding reliably in everybody, but rather, are there regions that are responding more reliably in certain groups of subjects, and does that vary at all with trait paranoia? So in other words, if you and I are more similar in our baseline paranoia, we might have a more similar interpretation of the story, and therefore there might be certain brain regions that are more synchronized between us, for example, than for someone else that has a different level of trait paranoia. So that's the analysis framework that we're taking for this particular study. And before I show you the ISC results stratified by trait paranoia, I'll just show you ISC across the whole sample, just as a sanity check. So And this is very much replicating work that are results that others have found using these types of stimuli. So essentially, if you just compute this synchrony index as people listen to the story across the whole sample, you see strong synchrony, for example, in auditory cortex, happily, because this was an audio delivered story. But interestingly, you also see a lot of synchrony in these higher order regions as well. And my favorite thing about this map is that if you look at some of the peaks outside of primary auditory cortex, for example, this peak in the temporoparietal junction or sort of superior temporal, posterior superior temporal area, and you do a neurosynth or a meta-analysis term search for this region, you see things like theory of mind and mentalizing. Same thing for this cluster down here in the cerebellum. So this is sort of reverse inference, but it's a bit of a hint that our story is provoking the types of cognitive processes that we were hoping it would in our subjects. But again, this is ISC across everybody, and we were interested in where there might be more or less synchrony according to people's levels of trait paranoia. So the next thing we did was to stratify people based on high versus low. And again, this was based on a separate trait level measure that didn't have anything to do with the story, that was collected separately. And what we see here, if we do a whole brain contrast, is that a few different clusters emerge as more synchronized in pairs of high paranoia participants as compared to pairs of low paranoia participants. And these regions include the left temporal pole. I'm showing you here the neurosynth or the meta-analysis terms for each of these regions. So we see left temporal pole, we see right medial PFC, we see a dorsomedial region in the PFC, and we also see left precuneus. And we thought this was really interesting because, again, everybody's getting the exact same story. We also did a lot of careful checks to make sure that there were no differences in performance on the comprehension question. So it wasn't necessarily the case that the high paranoia individuals were just paying better attention. There were no demographic differences between the two groups, so age and sex were pretty well matched. There were no differences in performance on the general cognitive batteries. So it really seems like there's something about the intrinsic paranoia level that people are bringing to the table that cause people that are more paranoid to be sort of more synchronized with one another while they're listening in these four regions. And the really cool thing about naturalistic tests is that you can then, so given these results that were acquired in a very whole brain data-driven way, we can then go back to the stimulus itself and see if we can figure out what events in the story were actually driving activity in these regions. And we're doing so more for the people with high paranoia than the people with lower trait paranoia. I'm going to skip this real quick and come back to that. So because we had crafted the story, we had actually sort of inserted some events that were deliberately meant to arouse suspicion, maybe in some people, but not others. And so what we did was we had a set of independent raters go through and mark events where the main character was either experiencing an ambiguous social interaction or was explicitly mentalizing about the beliefs or intents of other characters. And if you do that, there's pretty decent inter-rater alignment. So we were able to create just a sort of more standard traditional regressor out of this. So these are the moments that might arouse some suspicion in some people, but not others. And just to give you sort of examples of what each of those categories might look like. So this was an example of a socially ambiguous or mentalizing sentence, and this would be sort of a control sentence. So you can read these briefly for yourselves. But we took this regressor and ran a very traditional general linear model-based analysis. And we did this in an ROI-based fashion. So we had our two ROIs that were, if you recall from a couple slides ago, were more synchronized in the high paranoia group. And these were the ROIs that we know from previous work are involved in social cognition. So left temporal pole and right medial PFC. And we hypothesized that these regions would be more responsive to these mentalizing events in high paranoia individuals. We also had two control ROIs. We had a positive control in the left PSTS. This was a region that, if you'll recall from the very first ISC slide, was strongly synchronized in everybody, but didn't show a group difference. So we hypothesized that this region would be strongly reactive to these events in both groups, but again, no difference. And we also had a negative control region just here in primary auditory. So this region should not care really one way or the other about these events in either group. And essentially, when we ran the GLM and compared the beta coefficients, that's exactly what we found. So we found that in both the left temporal pole and the right medial PFC, people with high trait paranoia had more reactivity to these events in these regions. In the left PSTS, we see that both groups are strongly reacting in this region, but there's no group difference. And in left Heschel's gyrus, we see no significant reaction in either group. So, So this is just an interesting, you can start, with naturalistic tests to try to tease out not only where you're seeing differences, but why you might be seeing those differences, or what is the content that's actually triggering those differences. For reasons of time, I'm going to skip this. We did measure sort of behavioral reactions to the story using a free speech prompt, and we found some interesting signatures of trait paranoia in the types of words and semantic content that people used when talking about the story. Happy to come back to that if people are interested. But I do want to get through this last section, which is quite short. But just to sum up the second section here, so can we design a bespoke naturalistic stimulus to draw out variation of interest? This was a fairly small scale study, but I think it's a really interesting proof of concept that you can use a sort of intentionally ambiguous social narrative, for example, to evoke differences along a paranoia axis. And you might imagine doing this for other types of traits. So things like anxiety or maybe other personality traits, kind of having a stimulus that's tailored to that construct that you'd like to study, and being able to get a much more nuanced understanding of what types of information might be evoking differences. For the final section, how should we model individual variability during naturalistic stimulation? This is an issue I've thought a lot about because a lot of the analysis techniques that were designed for naturalistic stimuli, so things like intersubject correlation, which is such a beautiful and elegant approach, a lot of them were originally designed to capture group level phenomena. So what's common, what's shared across brains. And when we try to adapt that to study what's different across brains, we run into some interesting sort of theoretical statistical issues. And so I've been thinking a lot about this over the last few years and trying to come up with a simple but still powerful way to adapt those techniques to look at individual differences. I'm sort of calling this idiosynchrony. So a little bit of sort of quantitative stuff here. So Sam Nastase et al. Had a really nice paper in Scan a couple of years ago, kind of outlining the overall technique of intersubject correlation. And so in this framework, basically we can understand a voxel time course, which is represented by xi of t here, by a shared group component that's consistent across people. So this is the C, so the I here is denoting subject. So for subject I in voxel X, that time course is going to be composed of this C, this consistent time course that's shared across everybody. And then it's also going to be composed of these two terms that are specific to the subject. So one of them is this idiosyncratic response. So this is a response that's driven by the stimulus, but it's unique to each person. So if we try to just use basic intersubject correlation, we're not going to capture that. And then there's also potentially this error term here, which is anything going on in the subject's brain that's not actually driven by the stimulus. So this could be other neural activity that's just kind of mind wandering or, not stimulus driven. It could also just be physiological noise, scanner noise, lots of other stuff. So the point is that with traditional intersubject correlation, we really only have access to this term, or we're really only able to estimate this term here that's consistent across the whole group. But we know, or we think that there, might be a lot of idiosyncratic stuff going on that is driven by the stimulus, but just isn't consistent across all of our subjects. And so how can we try to separate these two terms from one another? One way we could do this would be a test-retest study where we see what's consistent within a subject across different viewings of a movie. And there's been some really interesting work along these lines. Of course, we also know that the experience of watching a movie a second time, especially sort of rich, engaging movie, might be different. So one other thing that we could try to do here, given, assuming we only have one repetition of the stimulus for each subject, is we could try to anchor this id term here, or this ID term, to something else that we know about each subject, like behavior. And this is sort of just a schematic of what this might kind of look like. But if we have a behavioral score that's known for each person, this is kind of similar to what we were doing in the paranoia study, we might be able to estimate this term as a function of that level of behavior. So again, the intuition here would be that there's some kind of canonical response associated with this behavior, and the degree to which people express that response is dependent on their level of behavior. And so this is a framework that we've been thinking about. And just to give you a little more intuition behind how this might work, the main issue that we're dealing with is how can we relate behavior, where we have one measure per subject, to ISC, where we're having fundamentally one measure per subject pair. You can't calculate ISC just for a single subject because it's inter-subject correlation. So you have this measure that's operating on the pairwise level, and you're trying to compare it to this measure where you have, just a single measure per subject. And the framework that we've come up with and other labs kind of converge on this at the same time, but we're calling this inter-subject representational similarity analysis. And if you're familiar with RSA, it's basically, it's just an RSA where your units are subjects. So basically what this looks like is for a given brain region, you can calculate a brain similarity matrix. And you can think about this like inter-subject correlation, although it could also be really anything else that you might extract about brain activity during the naturalistic stimulus. And you just calculate a subject by subject matrix as to how similar two people are in their brain data. And then what you want to do in the RSA framework is to compare this to a similarity matrix of behavior. And this may sound straightforward, although, and then, you would correlate the upper triangles of these and essentially brain areas where you see that the pattern of brain similarity matches the pattern of behavioral similarity. You might infer that whatever behavioral trait you're looking at is having an effect on your brain data. So this sounds great, although it turns out that calculating this behavioral similarity matrix is maybe a little bit more complicated than you might think. So for example, in cases where you have just sort of one scalar term per subject, so like let's say it's age or a summary measure of trait paranoia, how do you get a similarity metric between two single numbers per person? The traditional approach to this would probably be to look at Euclidean distance. So in other words, just how different are two people's behavior scores. But I think that there are actually many other potential ways of modeling distance that might actually be more accurate or more true to the way that we actually observe these phenomena. So basically what I just mentioned here, so these are all simulated data. These are toy matrices. I wish the real data looked this pretty, but these are just different ways of constructing behavioral similarity matrices. And so as I just mentioned, the sort of typical way to do this would be to look at Euclidean distance, so just how different the two scores are by subtraction. And but we're sort of, by doing that, making a strong assumption about what we think similarity actually means. And to illustrate that, what I'm showing you here is, this is again a toy matrix, which is subjects by subjects. I'm arranging subjects by their theoretical behavioral score from low to high. And so by doing this simple subtraction approach, we're essentially assuming that subjects that are similar in terms of their scores, regardless of whether they score low or high, should be similar in terms of their brain response. So in other words, if I have a scale that's measured from 0 to 100, this model is assuming that, if you and I score 0 and 1 respectively, we're going to look just as similar to one another as two subjects that score 99 and 100, because all that matters is the difference between scores. It doesn't actually matter where you fall on the absolute spectrum. And so this is what a Euclidean distance model is going to assume. However, what we started to see hints of in the Paranoia data set, and what we've seen strongly in other data sets as well, there are other possibilities for what this structure might look like. So for example, it could be the case that everyone who scores high is kind of clustered together, and then everyone that scores lower is spread, the variance just kind of spreads around them. So what this type of model would be telling us is that there's a strong canonical response associated with one end of the spectrum, for example, the high end. So pairs of high scorers are similar to one another, but low scorers are just sort of more idiosyncratic. They don't have a strong shared response, and they're just kind of spread out around these high scorers. And the Euclidean distance model is not going to capture that. So to capture something like that, you could take, for example, a mean between the two subjects' scores. And there's a few other ways to operationalize this same sort of structure where you have this strong shared response at one end that kind of spreads out as you move down the spectrum. And these are all just sort of showing different ways of operationalizing that. In this case, you might have this strongly shared response, and then There's a general increase in variability as you move down the spectrum. It doesn't sort of randomly go. It is still going in something of a consistent direction, but it's just much more variable. And so all of these different distance models, you basically have to choose one of these and make an assumption when you're doing this type of RSA approach. And so what we've been doing is we've been calling this this model the nearest neighbors model, because it basically just predicts that whoever you're most similar to on your score, that's who you look most similar to in the brain data. We've been calling these models the Anna Karenina models, after the famous opening line of that novel, which I think goes, all happy families are alike, all unhappy families are unhappy in their own way, but you kind of get the idea. So in this case, it would be all high scores are alike, all low scores are low scoring in their own way. And of course, that could be flipped around as well. So that's just sort of a detour into the types of modeling approaches that may seem kind of nuanced in the weeds, but actually make a huge difference, because if you're only ever using this model, you're not going to be capturing these types of effects that might be present in your data. And so we've seen several examples of Anna Karenina effects in the wild. This is a slide I skipped over from trait paranoia, but everything I showed you before was dichotomized kind of median split results. But it turns out that if you preserve the continuous value of this trait, you can start to see that in this left temporal pole region, as you move down into the left, these are pairs of subjects with increasingly high paranoia in both subjects in the pair. You see stronger ISC there, and there is a significant linear or monotonic relationship here. We're also seeing, this is work in collaboration with Alyssa Bayek and Carolyn Parkinson at UCLA. They're finding in a separate data set that more popular individuals have more similar neural responses to one another while they watch movies, while less popular individuals are more idiosyncratic in these regions here. And there's also, excuse me, this interesting paper that came out recently looking at schizophrenia patients compared to healthy controls, and looking at the sort of typicality of time courses, such that the healthy controls have a typical time course in response to a movie, whereas the patients with schizophrenia are just much more variable. And you can actually see in this two-dimensional projection that exact same pattern. So blue are the healthy controls that are all similar to one another. And then there's just this spreading out and just this much more variance in the patients. So I've gone longer than I was hoping to, but essentially just to wrap up this third section, how should we model individual variability during naturalistic stimulation? I think intersubject RSA is a promising framework, but we need to think carefully about our distance models. And it's not to say that one of those models is always going to be right. Usually we test a few different models in each region. So it could be that one model is best capturing relationships in one brain region or in one type of population. But these are the questions that we've been thinking about. Let's skip this for reasons of time. Our next steps, we are playing around with a data set that has 4 different independent films, and asking which film sort of best pulls out differences related to symptoms of mood disorders. And we're quantifying lots of different features of these films along the process, along different places in the processing hierarchy. So things from low-level features like loudness, frequency, luminance, flow. We can get semantic categories, and also high-level features like language and emotional tone. And one prediction that we have, again, this is simulated data here, would be that people might be more similar in their responses to these low-level features, but as you kind of move up the chain, you start to see more variability in reactions or in essentially evoked activity to these higher level features that we can pull out. So with that, I'd like to thank all my collaborators, both at NIH and Yale, and my new lab here at Dartmouth, my funding sources, and leave you with this final image of the dangers of averaging data across subjects.